Hey learners, welcome to Ahmad Coaching and I'm your teacher Dr. Anam. Today we will study about molecular carrier vector. When we talk about molecular carrier, it means that something which is going to carry but at the molecular level, the level of cell, the level of protein, carbohydrates. So we are talking about some things which can carry our desired things in the cell or at a molecular level. So the molecular carriers are actually vectors. Vectors may be anything, but in case of molecular biology, the vectors, they are actually DNA molecules that is used as a vehicle to carry a particular DNA segment into the host cell as part of cloning or recombinant DNA technique. It means that when you are cloning something or when you are using a recombinant DNA technology, you need to send your desired DNA into the host cell. So what you are going to do, that you are going to attach your desired particular DNA with another DNA which is actually going to work as a vehicle, or you can say that it is like a bike or like a truck, and you have loaded your DNA into it, and now that truck is going to take your DNA into the host cell. So that truck is actually your vector. The vector typically assists in replicating or expressing the insert DNA sequence into the host cell. So as it is going to take your DNA into the host cell, it is going to help in the replication. Replication means that one molecule of DNA is going to be duplicated, or it is going to help in the expression of the DNA, means that DNA is going to form protein. Now the vectors, they can be plasmids or they can be viruses. Talking about plasmids, the plasmids, they are extra chromosomal circular DNA molecules which are naturally present in the bacteria. Here you can see this is our bacterial cell. Here you can see this is its bacterial chromosomal DNA. While along with this DNA, there are some circular DNA present in the bacteria. These circular DNA are known as plasmids. They are not present in the chromosome, that is why they are called as extra chromosomal, and they are present in the circular form. They are present in the bacteria, and the important thing is that these plasmids, they carry genes for the antibiotic resistance as well as for the fertility. Antibiotic resistance means that if you place this bacteria on antibiotic, or you want to kill this bacteria with the help of antibiotic, the gene present in this plasmid is going to help the bacteria survive that antibiotic. So this bacteria is antibiotic resistant because of the plasmid, or because of the gene present in the plasmid. Plasmids were first discovered by the investigators when they were studying the sex life of the digestive tract bacterium Escherichia coli. So they discovered that Escherichia coli has this plasmid which is related to fertility. We can take the example of plasmid. We have this PSC101. This is the name of the plasmid, and here the P stands for the plasmid. So this plasmid has antibiotic resistant gene for tetracycline. It means that this plasmid has the gene of tetracycline, and any bacteria having this plasmid will be resistant to tetracycline. Similarly, another plasmid is PBr322. Here again the P stands for the plasmid and BR322 is its name. It has the antibiotic resistant gene for tetracycline as well as for the ampicillin. So the bacteria containing this plasmid will be resistant to tetracycline as well as the ampicillin. So how actually plasmid helping in the cloning? Why you are using it in the cloning, and how you can identify which bacteria has plasmid or your desired gene? So what you are going to do, you are going to place a gene of interest in the tetracycline resistant gene of plasmid PS101. For example, here I had the bacteria. From this bacteria, I'm going to isolate this plasmid. Here I have this plasmid. In the plasmid, I have this antibiotic resistant gene. For example, this plasmid is PSC101. So this antibiotic resistant gene is going to be tetracycline. What I'm going to do, I'm going to cut my this plasmid with the help of restriction enzymes from this part, and then I'm going to place my gene of interest or my DNA into this plasmid. So here you can see that I have placed my gene or DNA into the plasmid, and after this ligation I'm going to place my this vector or my this plasmid into the bacteria, and then I'm going to allow this bacteria to grow on a plate or on a medium which has tetracycline in it. So this is a plate, and on the plate I have this agar medium. Agar medium is actually a solid medium which has all the nutrients for the bacteria to grow. So when I'm going to place my this bacteria on this agar medium, the bacteria is going to use that nutrient and going to divide. This medium also has tetracycline in it, so only the bacteria which has our this plasmid with this antibiotic resistant gene is going to survive the tetracycline, and the rest of the bacteria which don't have this plasmid or antibiotic resistant gene, they are going to die. So you can see that only some areas, or you can see these small circles, they are actually the growth of the bacteria having our desired vector, while the rest of the area is clean because the bacteria present here, they didn't have our vector. That is why the tetracycline has killed them. So in this way I have decided that my bacteria having plasmid or my gene of interest is present in this part, or these are the colonies of that particular bacteria. So I can take the bacteria from here and I can grow it to study my gene of interest. So in this way the vector is going to help transfer my gene of interest into the bacteria, and then helping the bacteria to identify that which bacteria has my gene of interest by the help of antibiotic resistant gene. The second thing that we can use as a vector is virus. The virus, they are also used as a molecular carrier to transfer the gene to the host cell. The examples of the viruses which can be used as the molecular carrier or the vectors are retrovirus or adenovirus. What happened that we know that virus has this genome inside it. So what we are going to do, that we are going to place our gene of interest into the genome of the virus. So here you can see this is our gene of interest, while the red part is viral DNA. After that we are going to allow this virus to infect the cell. This can be bacterial cell or this can be any other cell in which you wanted to transfer your gene of interest. The virus is going to infect that cell and then going to enter into the cell. After the entry, it's going to release its DNA, and this DNA is going to move upward and then going to express or going to replace the infected copy, or whatever purpose it has to do, it is going to do like that. So this is how the plasmid and virus, they both are working as molecular carrier vectors and helping in the transfer of gene of interest into your host cell and then expressing or replicating your gene of interest. So this was our lesson on molecular carrier vector. If you still have any question related to this topic, you can ask me in the comment section. If you like this video, share it with your friends and also subscribe to my channel. I will see you in the next video.